All right, on this episode, human language gene inserted into mice led to some bizarre effects.
In that somewhat unusual experiment.
If you're
Of
using
course, it was gonna give me a
but
commercial.
don't know how to monetize it, watch this video to the end.
No, I'm good.
All right,
Experiment and experiment.
Okay.
Hello, info person. This is Anton, and today we're going to discuss a somewhat intriguing yet somewhat unusual experiment. An experiment involving mice that were genetically modified to have one single gene to be exactly the same as the one inside humans. And this produced results scientists are still trying to understand. Here, this gene that generates a protein known as NOVA1 may potentially explain why humans can communicate the way we communicate, and might have created our ability to produce complex ideas and to verbalize a lot of our thoughts.
That's interesting, bro. So now we're gonna have the ability to make animals be able to talk, like in Dr. Doolittle. Huh. That's interesting. Like genetics, bro. We're gonna change it to where like animals walk upright or something, like lion, cat, humanoid. The best I can put it right now.
And so let's talk about this somewhat unusual but somewhat exciting experiment in a little bit more detail and talk about some of the potential ramifications. Now, as always, you can find the study itself in the description below, and here the study by
Humanized NOVA1 splicing factor alters mouse vocal communications. NOVA1, a neuronal RNA binding protein expressed in the central nervous system, is essential for survival in mice and normal development in humans. A single amino acid change, I197V, in NOVA1's second RNA binding domain is unique to modern humans. To study its physiological effects, we generated mice carrying. We generated. Do you see what I mean? Do you see how crazy that sounds? They just generated mice like this out of thin air and shit, carrying the human-specific I197V variant NOVA1 Hue/Hue, and we'll see what else he's saying.
Yoko Tajima basically focused on a somewhat unknown protein known as NOVA1. It's a protein that binds to RNA molecules in...
Okay, so look, so yeah, see, I knew it was gonna have to do with CRISPR, see? But may it be in a... Oh. Proteins, no, this gene encodes in your specific binding protein, member NOVA family. Paraneoplastic disease antigen. It's based on a disease, bro, that is recognized and inhibited by paraneoplastic antibodies. These antibodies are found in the sera of patients where paraneoplastic opsoconus ataxia, breast cancer, and small cell lung cancer. Alternatively, spliced transcripts encoding distinct isoforms have been described. Both Neanderthals and Denisovans had one version, and nearly all modern humans had another, suggesting positive selection. Insertion of Neanderthal gene variant of the neuro-oncological ventricle antigen 1 NOVA1 gene into human cortical organoids might promote slower development and higher surface complexity in the brain models. Yet this may be an artifact of CRISPR side effect, as it could not be replicated in a subsequent study. Interesting.
Encoded by the NOVA1 gene, and in essence sort of looks like this. And though like many other genes and many other proteins, it seems to be responsible for a lot of different functions, Inside the brain, it seems to be responsible for neural development. As a matter of fact, all the
Okay.
previous studies about this gene were mostly studying this gene because it seems to be associated with a lot of different brain disorders, specifically when it has some kind of a problem. But by itself, this NOVA1 gene is also known as master gene regulator. You can essentially think of
Okay,
it as a kind of
that's crazy.
a master switch, and so
Okay, cool. That's all right.
any
Interesting.
problem with the NOVA gene results in some serious issues. You can actually learn about this gene and its function
NOVA1 is a master regulator of alternative splicing in pancreatic beta cells. Bro, gene splicing is getting crazy.
from one of the studies from 2014 that you can find in the description. But surprisingly, it does seem to affect at least 90% of all human genes in some way or another. But most of the interest in the last few years was really in regards to its effects on the brain, because scientists discovered that when it creates this NOVA1 protein, this can then rearrange sections of RNA inside neurons, making them form in a very different way, making them create certain structures and develop in certain ways.
The return
I
of
paused
Area
it.
51.
I
being
paused it
one of the
right
most
in time for a
high-performance
commercial.
desktop
Get out of here.
Here's another study
That's crazy. Okay, so what are you saying is like they can introduce. Bro, that's okay. Dude, they're gonna like I don't even know how to describe what I'm
from
watching.
just 2 years ago that essentially discovers that it seems to regulate very specific activity inside hypothalamus, with any change to this protein affecting our brain and hypothalamus in somewhat serious
Which
ways.
regulates your
And so
temperature.
The overall conclusion from many of these studies is basically that this unusual gene changes how our brain cells seem to synthesize proteins, and thus changes how our neurons develop. And one of the main conclusions here is that it seems to dramatically increase molecular diversity inside neurons. And naturally, because this gene is so important, it does not just exist in humans. As a matter of fact, it looks like most mammals, or possibly even all mammals, seem to contain NOVA1 inside their genes. And obviously, because this is a master gene, this should not come as a surprise. It would explain why so many mammals develop in very similar ways and even have somewhat similar brain structures. Except that there's a very small side note here. Of all of the mammals in nature, looks like human NOVA1 gene has one tiny difference. It literally has a single amino acid that's different in human version compared to other mammals. Here, inside the position 197, the isoleucine amino acid has now been changed to valine, which though might not be a big deal, completely changes the protein in humans compared to other mammals, at least in terms of its structure. And because of this bizarre difference, for many years now, some researchers speculated that maybe this is actually something that makes us very different, and specifically, maybe this is something that gave us speech.
That okay, so that's okay. So this very specific, like, so because like they're gonna look through the technological advances, the ability to map out the DNA strand is going to be immensely start happening. The only issue is with that is the system only can go so far down that hole because once you get to a certain point in the DNA, you recognize that all of the system is a lie.
So for the past few years, NOVA1 has actually been the so-called language gene contender because only humans seem to have this specific form. But this was somewhat debatable because previously, in a lot of different studies, a slightly different gene was a better contender. A gene we see right here known as FOXP2.
FOXP2, that's a crazy gene, bro.
Or fork-hat box protein P2, a gene expressed inside the brain, the heart, the lungs, and the digestive system that also exists in a lot of different vertebrates. And
Okay.
interestingly, one of the experiments back in the days revealed that when this gene is introduced into mice, they actually start to speak differently, or technically squeak differently. This was reported back in 2009.
Okay.
And here the human
All right, are you gonna play? Please tell me you're gonna play. Please tell me you're gonna play some mice talking, squeaking different, dude.
version of this gene made the mice produce slightly different sounds. Although here it also produced some additional bizarre behaviors. For example, the mice were not as curious anymore and did not feel as much joy because for some reason their brain was not producing as much dopamine. Nevertheless, this experiment and the study was actually exciting just because this was achieved and because it was now possible to test other genes just to see if we can discover this language gene and communication gene that in essence makes us human. Although when it comes to this FOXP2 gene, based on the analysis of Neanderthals and Denisovans, or essentially our cousins from back in the days.
Right. Okay. Lots of information, bro. That's bro. So like, if they figure that out, bro, if they figure out what makes like why I'm doing what I'm doing right now, which I comprehend is consciousness, but I'm pretty sure there's some biological counterpart to allow certain aspects of consciousness to be happening, like the ability to speak and communicate and do that sort of stuff. Yet I already thought that all like mammals, at least for what it's worth, have the ability to do what humans do.
They also seem to have
Other
this
than
gene as
talk.
well, and it was very similar to the one in humans, which back in the days implied that maybe Neanderthals
And defy nature. Like animals don't have to really. Well, actually, the animals do defy nature sometimes. That's interesting. I didn't think about it like that. Okay, cool. Yeah, see, awesome. Oh, I hit the mute button.
One of the reasons back in the days also seemed to have this gene as well, and it was very similar to the one in humans, which back in the days implied that maybe Neanderthals could also speak like humans. And one of the reasons scientists actually thought that FOXP2 was a language gene was because of the disorders associated with the problems inside this gene. Anyone that has a mutation in this gene very often has a lot of speech defects and is unable to move their lips and their mouth in order to produce any speech.
Wow.
Likewise,
Okay.
compared to
Cool. I
other
don't have
animals,
that.
the human
I'm glad I don't have that issue. I don't know what that issue would be. Talking a lot. I talk a lot.
FOXP2 gene is just a little bit different, but is exactly the same as the one inside Neanderthals. And so this was thought to be the origin of human language. But naturally, this was just the beginning, and obviously scientists wanted to keep looking just to see if we can find other contenders or discover something else.
Right.
Had fallen victim to a scam incident.
A victim of a
Yep.
And
Get
so
out of here.
6
I'm
years
not a
later,
victim of a
we get a new
scam.
study. A very similar experiment, but an even more exciting discovery. Because in this case, this Nova1 gene is definitely unique to humans. Neanderthals and Denisovans did not have the same gene either, and it seems to be one of the few genes that pretty much all humans contain. Now here I'm saying almost, because there is a very bizarre discovery. Researchers were able to collect anonymous DNA sequences
Right.
from approximately 650,000 humans
Damn.
and discovered this gene in
That's a
almost
lot.
everybody except for 6 people. Now because these were anonymous, we have no idea who they are, but for some reason, out of 650,000 people, 6 contained a different variant. Obviously there's no explanation why and why they have this mutation,
crazy.
but it would be interesting to find out if this produced any kind of a problem with their speech. But based on the genetic analysis, we know that this gene is relatively new in terms of evolutionary times. Since Neanderthals and Denisovans did not have this gene, it must have evolved after the species split. And based on the
Okay,
thorough
damn.
genetic analysis, scientists believe that it must
The Matrix. Look.
have formed only a few 100,000 years ago, possibly even less. And so because of the novelty of this gene, it was obviously somewhat intriguing to discover what effect this has on humans and what effect it might have on these mice. And that's because mice, despite containing a similar gene, do not contain the same gene. This is really uniquely human. And to test this, scientists created genetic mutations that made these genes exactly the same as human genes, and then conducted 2 separate experiments, one involving pups and one involving grown-up mice. And even as pups, these mice were already doing something different. Compared to a regular pup, these genetically modified pups were producing frequencies that were much higher. Basically ultrasonic squeaks, but in much higher frequencies than usual. And for some unknown reasons, despite a lot of screaming, their moms seemed to ignore them.
What the fuck? That's crazy.
Normally, when pups produce ultrasound squeaks, their moms come right away. But here, even at young age, these pups were already producing different sounds and thus communicating a little bit differently. But a much more intriguing discovery came from the mice that were already older. So basically, once these pups grew up and became older, they then went on their first date, or technically had their first courting experience. And surprisingly here, during courtship, these genetically modified mice were producing calls that were very different from a regular mouse. And though the frequency was exactly the same, the actual amount of squeaks was extremely different and much more complex. In other words, they were actually making sounds that were way more complex in terms of syllables with the overall communication being longer and more diverse. In other words, I guess in human terms, their pickup lines were very elaborate.
Oh, no, I've scared you. I've said too much. I'm hopeless and awkward and desperate for love.
That was a great plug-in. That was a great plug.
And this is a
Fucking Chandler.
really bizarre discovery. It really confirms that the mutation in this NOVA1 gene is potentially responsible for our ability to communicate with so much complexity and our ability to express so many complex thoughts in so many different ways. And because here this gene could also be tracked inside the brain of the mouse, it definitively confirmed dramatic changes in a lot of different sections, especially the ones responsible for communication. And this was a direct and definitive confirmation that NOVA1 protein seems to dramatically change vocalization patterns and may indeed be the human language gene, a human-specific gene that literally built our culture. And these bizarre vocalization patterns that were detected in these mice, though preliminary, still seems to suggest that even in these mice the communication became way more complex.
Bro, so like they were able to test the difference in regular mice versus like squeaks. The little sound, like that's it. So like basically there's a mouse language now. They figured out the mouse language.
And much more.
They don't know what they're saying. They just know it's based on like shorter, longer, or more or less squeaks.
Elaborate. But unfortunately it seems to have failed to impress other mice. In other words, just having this one mutation would obviously not suddenly turn them human. And since the brain analysis confirms that there's a change in the vocal behavior area, right now there's little to doubt that NOVA1 was indeed responsible. But in humans, it obviously does so many other things. For example, we know that it also affects learning, and problems with NOVA1 have been correlated with various learning disabilities. And that's on top of approximately 90 other things it seems to be responsible for.
Okay,
So
dang, it's
here,
responsible
it
for a
might
lot.
not be just a language gene, it might be a gene responsible for a lot of other things that make us human. And something that very likely evolved in early humans and eventually became dominant because it very likely provided a lot of advantage. Advantage in communication, but also possibly things like learning and even understanding the environment. But because this is just the first of such studies, we still have no idea exactly what it does or what other effects it might have inside these mice that were now basically humanized, as the researchers refer to it in this study. And so I'm actually kind of excited to see other studies of these humanized mice, because here this discovery might finally show us the actual genetic mutations that transform.
Alright, yeah, I gotta stop there because like he keeps showing this little chart behind him, and unfortunately, bro, ones didn't come from monkeys, bro. Not the ever like that's not how it works, bro. So I don't know, or maybe I'm not reading this chart right. But if you want to go check, it was still a great video. If you want to check it out, go check it out. Anton Petrov, human gene, language gene inserted device. I'll put it in the chat. Just in case anybody wants to go watch it later.